All right, so now we're going to move on and start talking about some of these phylums. So this lecture is going to cover quite a few. So we're going to talk about the Placozoans, your sponges, your comb jellies, and the Cnidarians. Hopefully I'm saying some of these right. If I butcher names, I am so sorry, but we're going to cover quite a few phylum in this lecture. And I tried to keep them in small groups, and you see some similarities why these things are grouped the way they are. So this is an example of a sponge right here. If you saw that, if you're a scuba diver, this is actually an animal. People will probably think this is plant, but this is actually an animal. Quite fascinating, I think. So as I mentioned, you're going to see this show up at the beginning of a lecture. I kind of like it because it kind of talks about why things are divided the way they are. Now, one way that we informally divide things is if we call it an invertebrate versus a vertebrate. And it has to do if it has a backbone or not. And you're going to see when we talk about all the different parts of the animal kingdom, 95% are invertebrates. Probably some of your favorites are going to be vertebrates, but 95% of all the animals are invertebrates. Quite cool. Now we are going to talk about the simplest of animals. We're going to start at the very, very bottom right here. So what we're talking about is these four groups right here, and we're going to talk about ones that have no true set of tissues. And then we divide it up by if they have specialized tissues and how they go through their symmetry. So we're going to talk of those that have radial symmetry. So what you're going to see is we're going to talk about your jellyfish, your placozoans, your sponges, and then your comb jellies. And I'm going to go through each of them. There are some phylum that are going to have more information, some not as much. You're going to see sometimes the book only gives like a paragraph about some of them. I tried to find some interesting facts, but I'm going on based off what I could find too, and I didn't want to overwhelm you with too much information. So your first phylum is Phylum Placozoa. This little guy right here. Not that interesting, but it is an animal. So where are you going to find this? In your coastal marine communities, primarily in these tropical mangrove habitats. Now, these little guys are transparent. They're going to be flat and disc-shaped and asymmetrical. So there's no real way that you could draw a line right here and get like a mirror image. Now, it does have a distinct top and bottom. Guessing how this image is set up, that this would be top and bottom. I don't know if this is top and underneath is bottom. But what I find kind of cool about this is only 4 types of cells in this total organism. Now we'll have about 1,000 cells total, but there's only going to be 4 exact types. So probably this external layer, some internal ones, but it has no distinct tissues or organism. Very, very primitive animal. Now there's only one species, the Tryphoplax adheriens, that happens to fall in this. And if you want to pick this as your favorite invertebrate, you can. But you know, like I mentioned, there's not a whole lot out there on them. Now they are going to go through asexual reproduction via the process of fission or budding, but in extreme conditions, some of them have been shown to reproduce sexually. Now, how do they go through and eat? Well, movement is pretty much just floating around in the aquatic environment, but they consume algae via digestive enzymes to consume food. As I mentioned, not much on these guys. I don't know if the book even has them in there, but I wanted to throw them in there because it does show up on the diagram. Now, one that your book does love to talk about quite a bit is Phylum Porifera, which are your sponges. Now, there are about 5,000 classified species. What's interesting about those, they do have some specialized cells. So our previous one then only had 4 cell types, but we're going to start to see now we're going to start to get more specific types of cells that are going to have specific functions. Now, all of these happen to be aquatic, and most of them are going to find in the marine environments. Now, there is an asymmetrical body plan with this, so it's kind of hard to find like that mirror image, but most of the time you're going to see them this cylinder shape with that large central cavity, that kind of that mirror that much right here. And then they're going to have spongocyl, which is that central cavity, and an opening up top, which is called the osculum. Now, how these guys eat? They're going to be immobile, so they attach to an area, and then what they do is they go through this process called filter feeding. They are suspension feeders. So pretty much water just moves through the body and they filter the food through it. So here's a little bit more about the morphology of them. And like I said, some animals I will go through more on the morphology and the reproduction. Some I won't. Like I said, the book kind of is all over the place. So with that, you're going to have, first off, is the spongocyl, this large central vacuole cavity right here. Now, depending on the type of species will determine how large and the size and shape of that. Now, at the very top of that, you're going to have the osculum, which is just the opening at the top. And then with that, you're going to have what's called the mesophyll, sorry, I'm used to saying mesophyll, which is a jelly-like substance between the outer layer and the feeding chamber right here. So if you kind of look at that, it kind of looks like the spongy stuff right there. Now, if you kind of take a little area right here and zoom in on it, you'll see some of these different types of cell types. So you'll see spicules, which are going to be little protein fibers embedded on the mesophyll to give structure. And then you're also going to see choanocytes, which are going to be called collar cells, and they're going to have flagellum. And what these are do, they're going to ingest and package the food into vacuoles via phagocytosis. So remember, that's how we get things in and out of cells. So that's the process of absorbing it into the cell, so then you could be broken down later by digestive enzymes. Now, you're also going to have these amoebocytes, which take in vacuoles and nutrients delivered to the other cells that are going to be located in the mesophyll. So this is a diagram that goes and talks about some of these different little types of cell site, specific cells I was talking about. Now, as I mentioned, they are filter feeders. So what they're going to do, if you kind of look right here, there's these little channels, or these porocytes. And what it's do, the water is going to flow through it right here. Now, as the water flows through here, it's going to go through and trap and filter the food particles in these chondrocytes, and then that's going to be taken into the mesophyll to be absorbed. Now, some more physiology on these guys. They lack a complete digestive, respiratory, and nervous system. As I mentioned, these are your very simple, very primitive animals. Now, you're going to trap food as it passes through the ostia and through the osculum. And primarily, they're going to go through and filter a lot of bacteria, okay? They're going to be going for the smaller stuff that they can easily absorb into their cells. Now, they do have to break it down, so they're going to use intracellular digestions, but what's important is the food particles have to be smaller than the individual sponge cells so they can take it in. So here's a video that kind of talks through that process right here. Now, they go through the process of gas exchange, they go through circulation, excretion are done via diffusion through cells that line the opening. So it's not like they go through breathing, it's pretty much through simple diffusion. Now, when they're adults, they are sessile. They'll be attached to a surface. They're not going to be moving. Now, how do they reproduce? They go through asexual reproduction. So they can use different types. They can use the process of fragmentation where they just break a piece off. They can go through the process of budding where you'll have an outgrowth from the parent and it just kind of taps off. Or they can make these things called gemmules, which are cluster of cells surrounded by a rough layer, which I have shown right here. Now, these gemmules can survive these hostile environments and then recolonize at a different location somewhere else when the environment improves. Now, some of them can go through sexual reproduction. Some happen to be monoecious, where that means they're hermaphroditic, and where they can produce both eggs and sperm. And then they can change when they order they're going to reproduce. I'm going to produce my eggs first and my sperm later, which is kind of interesting. They can go through this process where they'll produce the eggs first in their amoebocytes and keep them in the spongocyl, and then produce the sperm in a different type of cell, the choanocyte, and then they'll go through fertilization. Really kind of interesting. I don't know people how they study this, but they do. Some people love to study sponges. So let's move on and talk about, probably a lot of people love these, this Phylum Cnidaria. So this is going to be your jellyfish. These are so fun to watch. You're also going to have your hydra, your anemone, and your coral. So about 10,000 species found there. You're going to watch a movie about coral bleaching when you get to the ecology section and how important coral is when it comes to food webs. Those corals, oh my gosh, that broke my heart. There's a special on Netflix I watched that just talked about how this group recorded coral bleaching over time, and it was kind of sad to see. Now you're going to find most of these in marine environment, but some do survive in freshwater. Now there's a lot of diversity when it comes to the morphology right here, and you're going to see some with radial and some with bilateral symmetry. Now the key thing when you think about these guys is they're going to have that cenocyte, which is going to be a stinging cell, and they're going to have this is going to be located around their mouth and their tentacles. So you're used to seeing this bell shape right here and these long tentacles, which are their stinging cells right there. Now they use it to help them catch a prey and repel predators, and it's going to have those democysts, which are stinging organelles that thread light, and they go through and kind of puncture through and kind of neutralize and keep their prey like in this suspended state so they can go through and consume them. So a little bit about the physiology. They have two tissue layers, the epidermis and the gastroendermis, right here, shown right here. And then they have this space called the mesoglia in between. But pretty much you're used to seeing like the overall bell shape right here. And if you look kind of underneath, this is where you're going to find the opening, which is the mouth or the anus. Now they do have some differentiated cell layers in each layer. They're going to have some nerve cells, or enzyme secreting cells where they can break things down, and nutrient absorbing cells. One key thing about these guys, there's no organs or organ systems. As I pointed out, they have that single opening for both the mouth and the anus, and when they take food in, it's digested via the enzymes, and then it's going to eject whatever it doesn't need to do. Now it does have those nemocysts, or those stinging tentacles, and what they do is they kind of go through and harpoon and poison and stun the prey. Now we are starting to get to that very simple nervous system. They're going to have a net of neural cells located in this head part right here, and it goes through and exchanges oxygen, carbon dioxide via simple diffusion between their epidermis cells and water. Now reproduction on these guys is kind of interesting. Here's a cool little video that kind of goes through this. I've used it before. I think it's kind of cool. Most of the time when you think of you guys, you think of them when they're in what's called their medusa state. But they go through this stage where they actually are in this sessile state. So when it comes to there's a two-stage life cycle, there's going to be a polyp stage right here on the bottom, and then the free-floating medusa stage. Now it can exhibit one form or both, or it depends, it can be both, only one. It depends on the type of species that you have right here. So we'll have initial fertilization happen, the zygote, and those larvae that'll eventually go into a sessile stuff, and it'll go through its changes, become a budding polyp. And then you'll see these polyps kind of just like release, and they form like these little baby looking jellyfish right there, and then those will go through and become bigger. Now what's interesting is the medusa stage will produce sexually via external fertilization, and the next generation is going to reproduce asexually via segmentation. So this video is really cool, and it goes through different stages. I like it a lot. So just a little bit about the diversity, some of the classes you see in this phylum. You'll see class Anthozoa. Like I said, I'm going to butcher some of these up really bad. These are going to have no medusa stage. Your sea anemones, your sea pinion, and corals fall into this group right here, about 6,100 species. And then the class Psychophozoans, they're going to be your jellies, and these are going to be more moldal and primarily in the marine, and you're only going to see about 200 species. So this is probably what you're used to seeing when it comes to the aquariums. So two more classes. We have class Cubozoa, which happen to be your box jellyfish. It's more of this box shape when it comes to the bell part. But the main difference is how their tentacles are arranged, and they're primarily going to be in their polyp form if you're looking for these guys. And these are some of the best images I could find representative of each class. And then finally, you're going to have class Hydrozoa, about 3,500 species. These can be kind of interesting. They have both the polyp and the medusa form, but they form these colonies that can be free-floating, contain both medusa and the polyps. Your man of war fall into this group right here. Now, the last phylum in this lecture, which I know it's a lot of words right there, is Phylum Ctenophora. I've grouped this with them because these are your comb jellies. So they have enough differences to where they warrant their own phylum, only about 100 species. And what's unique about these guys, I think they're quite beautiful when you look at them. They live in marine waters worldwide, but they're going to use these cilia on the outside to move, and they kind of beat in a synchronized manner. If you ever find a video of these to watch, they are very cool. And they also use tissue contractions to move. Now, like just their cousins over there in the jellyfish land, they do not have a brain or a central nervous system, but they are starting to have that nerve net. Now, with these guys, they lack stinking cells, so that's why they are not classified in the other phylum, but they use sticky cells called colloblasts to capture their prey. They're hermaphroditic. They go through self-fertilization, but they can also utilize fragmentation. Now, this is why these guys have some controversia. Controversia, it's a made-up word. When it comes to classification, there's a dispute. As I mentioned right here, some of them think they should be here because, you know, they have a pair of anal pores. But then they have this other layer of tissue, and it's like kind of, you know, controversial. As I mentioned, your book doesn't talk about these, so this is just a bonus phylum, some bonus information for you out there. And then to wrap it up, like I said, here's a review. I think it's a more fun videos. I tried to find some different videos about different organisms, and you might find some of these interesting. And then my credit to my pictures.